Zempleni, could you please share with us your journey and how your interest in milk exosomes developed?
Okay, but first of all, thank you for inviting me to have this interview as an editorial board member. Start off with my educational background. I obtained my undergraduate and graduate training in nutrition sciences at the University of Gießen in Germany. During my graduate time, I emphasized pharmacokinetics, which is the mathematical modeling of nutrient turnover and nutrient metabolism. I had this lifelong interest in molecular biology and biochemistry. So with my postdoc at Emory University in Atlanta in the mid-90s, I really took advantage of the opportunities provided and then really broadened my horizon and then picked up lots of molecular biology techniques and then biochemistry techniques before securing my own faculty position here at the University of Nebraska-Lincoln. That was in 2001. And milk exosomes and milk non-coding RNAs, that came about in 2013 when I had a student, a very good student, Scott Bayer, who had an interest in non-coding RNAs. And that's how we got into this field of vesicles and non-coding RNAs. Initially, the focus was on microRNAs, but we quickly, and probably the first nutrition group, to realize that there is more, has to be more than just microRNAs, and that's how we got into the extracellular vesicle field. And I have to say, the American Society of Exosomes and Microvesicles played an instrumental role in our transition from microRNAs to more broader horizon extracellular vesicles through the meetings, the annual meetings that the society hosted. Drug delivery, which is part of my research for exosomes, came about later when we got some funding to conduct drug delivery research.
I see. And we also noticed that your research on extracellular vesicle in milk, nutrition and drug delivery was recently recognized by the National Institute of Food and Agriculture. So could you please share with us any latest progress on that work?
Mm-hmm. Yeah, be happy to. I mean, I had various funding agencies support our research in extracellular vesicles. So NIFA, the National Institute, is one of them, but we were also supported or are supported by the National Institutes of Health, by foundations like Gates Foundation, Gerber Foundation, and the SYNGAP Research Fund, in addition to some industry sponsors that support our research. We have two pipelines, or two major pipelines of research. One of them is the nutrition angle, so milk vesicles in human nutrition, and the other one is the drug delivery angle. This recognition that you were referring to, this NIFA recognition during the National Dairy Month, came about because I typically keep very close contact and keep my program officials at NIH and the NIFA and other funding agencies very well informed of our progress. And during the National Dairy Month, they were looking for a person to feature on their blogs and tweets and so on, who is conducting innovative milk research, because USDA or NIFA is mandated basically to conduct food-related research, and that's why my name naturally came up. The progress, I can do that. Let's put it this way. I cannot give away any trade secrets in our lab, because we have some really high interest and high impact projects in the pipeline, as well as high impact papers in the pipeline. So first of all, I think I'm not much, and I think you mentioned biomarkers. We are not a biomarker lab. I mean, that's for you to do, but so I'm not the right person to comment on milk exosomes and biomarkers in general. For our progress regarding the other areas, I mean, nutrition and drug delivery, I'd say, yeah, we do lots of work in the microbiome exosome interaction arena, and we also invested heavily in milk exosomes and neuronal development. So these are two of the key areas that we do in the nutrition field. And in the drug delivery field, we really focus on loading milk exosomes with not just drugs or therapeutics, but also with CRISPR-Cas for gene editing and so on. So these are some of the key things that we are focusing on right now.
Actually, I'm also curious to know that what are the key differences between the milk-derived exosomes and bovine serum albumin-derived exosomes? What are the main differences?
So bovine milk exosomes and serum-derived exosomes, you say?
Yes, like BSA.
Okay, yeah. So we never did this comparison. We never worked with serum-derived exosomes from cows. I mean, we really limit ourselves to work with the milk exosomes. Serum-derived exosomes or urinary exosomes we use in some of our human studies, but that's really more when we look at the metabolism of these milk exosomes in our model organisms. And model organisms in our lab, in addition to the microbiome, is really humans. So we do lots of human research, and we do lots of research with mice.
I see. And what are the parent cell type from where these milk-derived exosomes are originate from? What are the main cell types? Can you tell us about that?
Yeah, that's a very good question. That's actually something that we also work on. Because I think it's not entirely known. There's a couple of papers out there suggesting that these milk exosomes originate in the mammary gland, but I think the evidence is fairly circumstantial. It's based on mRNA expression profiles that are similar in milk exosomes and mammary gland exosomes, or microRNA expression profiles that are very similar. But I think it's fairly circumstantial. So we really, at this point, we try to answer that question by using a transgenic mouse that we have developed. But yeah, so in all likelihood, I'd say it's the mammary gland, but we cannot exclude the possibility that some of the exosomes in milk actually transcytose and come from tissues other than the mammary gland.
So actually, if we know that, then we can produce these exosomes in vitro, right?
Correct. That is absolutely correct. And we do that in our lab. I don't know if you noticed our paper that we published last year with these bovine mammary alveolar cells that we're using. Yeah. And that's really cool, actually. So we have this cell line established in the lab. It's derived from bovine mammary glands. And these cells secrete exosomes that look exactly like milk exosomes as far as their cargoes are concerned, as far as their morphology and their size is concerned. And the beauty of the system is we can genetically engineer these MAC T cells and basically change everything that we want. There's surface proteins, cargoes, and then we can really use these genetic engineering technologies to interrogate milk exosome dependent pathways in humans or mice.
And so what do you think about the potential of future research with this milk originating or milk derived EVs? So you work with EVs, right? Not exclusively exosomes, because EVs, as you understand.
So in our papers, yes. So in our papers, sometimes we refer to these vesicles as exosomes because we feel like we established clearly that these exosomes or small extracellular vesicles carry all these markers that show they're of endosomal origin. So often we call them exosomes. Sometimes we call them small extracellular vesicles because they have the size of an exosome. But I don't think it really matters. It's just nomenclature, and we define in our papers what we have done, and then our readers and then the audience can decide if they want to call them exosomes or small extracellular vesicles. So the future, I think, like I said, we work in human nutrition and we work in drug delivery. Human nutrition, I think the focus probably has to be on infants, because infant is the only population group that exclusively relies or should rely on milk as nutrition. as you know, adults like you and I, many of us are lactose intolerant, don't consume any milk or cannot tolerate any milk. And so the pool of the affected persons is a little smaller. So infant nutrition, I think, is the way to go for human nutrition. For drug delivery, as I mentioned earlier, I think it goes really into loading these exosomes, not only with siRNA, that was the initial approach that people took to turn off harmful genes. It's really more gene editing, which is probably the next step. So load these exosomes with CRISPR-Cas tools, basically, and then edit genes in patients.
So you began the extracellular physical research around 10 years ago. Could you please share your vision and what makes you still adhere to this particular field?
Yeah.
Because many people have started working with cancer and neurodegenerative disorders.
Yeah, it's a good question, interesting question, actually. So you're right, we started this work in 2013. At that point, I was actually running a very well-funded lab with a focus on epigenetics and also related to vitamin metabolism, and consider myself a well-established investigator. And then I did something that goes against common wisdom, and I changed my research direction big time. I stepped away from epigenetics, stepped away from vitamin metabolism, and basically started all over in non-coding RNAs and extracellular vesicles. And again, that goes against common wisdom, because line of thinking is, well, if you're well established, why change the field? Just stick to what you have. But when we started in this microRNA and extracellular vesicle field, I really loved it. And I loved it for a number of reasons. First of all, it was completely new to us, and there was a steep learning curve. So we really learned a lot as we were going into that field. And it paid off funding-wise, so we have quite a bit of funding from various agencies. I also like that this topic appeals to really high-quality graduate students, undergraduate students, but also postdocs. So it really makes it, I shouldn't say easy, but it makes it fairly easy to attract very motivated and then high-quality people to our lab, to our team. Yeah, and what makes me adhere, you asked. I did mention earlier that we have a number of high-profile discoveries in the pipeline that have not yet completed or have not been published, and I definitely want to see them through to completion. So that's number one. Yeah, it's just excitement. I mean, it gets me excited, this field. So that's why I stick to extracellular vesicles.
Yes, this field is indeed interesting.
Yeah, it's growing fast too.
Yes, really fast. So what would be your advice for the new researchers who are maybe interested in pursuing a similar career path in EVs, particularly?
Yeah, that's interesting. So, I mean, first advice probably would be, and that's something I followed myself, I mean, just do what makes you happy. I mean, I told you about my change in focus mid-career or late career, and it was great. Even though normally people would say don't do it, but I think you want to do something that makes you happy. So you're very young, I suppose, and I think you don't want to get stuck in an area of research that doesn't get you excited anymore. So I think you really want to follow your heart and do what you like the best, even if it carries a little bit of a risk. The other advice would be persistent. I mean, back in the days when nutrition, especially in an extracellular vesicles, were somewhat controversial, or still is somewhat controversial. There's setbacks. I mean, manuscripts get rejected, grants don't get funded. So you just have to be persistent and then keep pushing. So then don't get discouraged, because you are working in a hot field, and then you will succeed eventually if you persist. And then for, let's say, students out there who may be watching this, or postdocs, if you want to get into the field of milk exosomes, contact me. We're always looking for highly qualified and highly motivated people to learn something new.
Okay. So have you ever checked the cargo contents or the differences in milk-derived exosomes from different species, like cow, goat, or even
Yeah.
human?
So we did not do a comparative analysis, but we have published a few papers in which we report on the microRNA, protein, and lipid content in human milk exosomes and in bovine milk exosomes. But we didn't really set these papers up in a way like we do compare human versus bovine. It's just you can look it up in our papers and can do the comparison of yourself if you want to, or the viewers can do that. All the data have been deposited in public repositories, so it's all accessible. But no, we have not sit down and then say, okay, let's compare what these things have.
Right. Okay, we would like to express our appreciation to Professor Zempleni for your presence and for your valuable research contribution to this particular field. It was great discussing with you the new direction in the field, and we strongly believe your future study will tell us in more detail about milk-derived EVs and their contribution to human health. So best wishes again, and thank you so much. And also thanks to the organizer for all your efforts in conducting this event. So
thank you. I really appreciate the opportunity to talk with your team.